Thank you very much, Lukáš, for that introduction. It's a great pleasure to be back in Prague after all this time. I did come here for the first time in 1989, and it was a very different world back then. I still have wonderful memories of that trip. It was quite exciting as an Australian at that point, visiting Germany when suddenly the wall came down and I came across to the Czech Republic, or Czechoslovakia as it was then, and then on to Berlin, where I had a pen friend, and I had met up with her a few months before the wall came down. And it's wonderful to be back here, and of course the country has changed quite a bit since I was here back in 1989. What I want to talk to you about today is the sort of a broad sweep of a couple of projects that have been recently conducted in my lab to address questions concerning the evolution of plant sexual system diversity and the function of males and females in plant populations. I'm not going to say too much about the evolution of sex chromosomes, although that is a major focus of much of our work here. But the question is sort of a broader one, and that has to do with the evolutionary transition between hermaphroditism and diesi, separate sexes. So I start with this slide here, which basically is there just to remind us of the incredible diversity that we see in flowering plants in terms of the strategies they have evolved to reproduce. These are all flowers or inflorescences, and in a sense it's a bit surprising that we see such diversity, given that actually what these flowers are there for, you would think, is simply to transmit genes to the next generation. They're all doing the same thing, and yet they've evolved so many different ways of doing it. And so this is something that's motivated at least some of our research. Why do these flowers have such different ways of basically transmitting genes to the next generation? The way I want to address this is actually maybe slightly unusual for a research seminar, because I'd like to consider this. Oops, I don't know why that is. Anyway, you can read what's on. A slightly unusual approach, because I'd like to consider this from the point of view of undergraduates learning botany and trying to understand how the scientific process works. And in that context, I'll introduce some of the questions that we've been interested in and some of the answers that I think we're getting towards through the work we've been doing. And so this slide sort of emphasizes 4 different aspects of the way we do science, I think, and it's something that we've tried to introduce to our students in field courses. So it starts with some sort of discovery or observation, which might lead to a particular question. And of course, in the end, one of the hardest things to teach students is how to ask questions. But that's what our business is as scientists, to ask questions, and it's probably the hardest part of science, working out what the most interesting question is. Then, of course, we need some sort of hypothesis or a little bit of theory, and it sort of helps to reflect on data and the assumptions. So that's sort of what I'd like to build on. And so there are going to be a few questions, simple questions that an undergraduate might ask, and how we might then go about addressing these from the point of view of plant sexuality. So let's start with an inflorescence of this Linaria species. And the question is, what's the function of a flower? What's the function of a plant, in fact? And there's a lovely quote that addresses this question in a book that influenced me a lot when I was a master's student in Australia, by John Harper, who was, I guess, the founding father of plant population ecology, who worked in Wales. And this is what he said: a plant is the only means with which a seed produces more seeds. So I've sort of called this the seed-centric view of plant ecology and perhaps plant evolutionary ecology. And I think it's interesting that he said this in 1977, which was 1 year after Richard Dawkins published his Selfish Gene, which is, an organism is only there for transmitting genes from one generation to the other. And so I think it's sort of interesting to speculate that perhaps John Harper was influenced by that particular perspective. But I think he missed something important in defining a plant's function in that sense. And that is, what about the males, or more generally, what about the male function? Because a plant is not there just to transmit genes through seeds, but also through pollen, if we're talking about flowering plants. And it turns out, and I think there's been a major change in the way plant evolutionary ecologists view floral biology since Harper's day, and that is the reincorporation of the importance of the male function to floral ecology. And I say the reincorporation because it was very clear to Charles Darwin that the male function was very important, and some of the hypotheses for floral dimorphisms that people are still working on today go back to Charles Darwin, who had a very ecological point of view, and even though he didn't know about genes, he certainly saw the importance of the male function of plants. And so that's an interesting question. What's the implication of the male function of a flower? It's not just producing seeds, it's transmitting genes through pollen. And this is where the little bit of theory can go a long way. And if we think about how to address this question, one way of doing that is to consider the strategies that plants have evolved in allocating resources to their two functions. So plants are not just producing seeds, they're transmitting genes through their pollen. There's a male function and a female function, and both of these functions are actually equally valuable. Every individual has a mother and a father. The male function is exactly equivalent to the female function in a sexually reproducing population, and so we should expect individuals to be selected to allocate equivalently to their male and female function in an outcrossing population, which is why we have a 1-to-1 sex ratio in so many organisms. And so we'd expect a 1-to-1 sex allocation to male and female function in hermaphrodites too. But that doesn't mean that the male and female functions are equivalent in terms of how selection is going to operate on them. So if we think about this particular simple figure here, where we're plotting fitness either through male or female function, this is the number of genes, for example, being transmitted to the next generation, as a function in this particular example of the attractiveness of the plant, in other words, the allocation of resources to attracting pollinators. And it might be measured in terms of the number of insects visiting an inflorescence, a flower or plant. And from the point of view of the female's function, a pollinator comes to a flower loaded with pollen, dumps a load of that pollen onto the flower's stigma, often many more pollen grains than there are seeds to sire. And so after a single visit, the female function may actually be no longer worth investing in further. And so we might expect this sort of fitness gain curve where it's worth investing a little bit to attractiveness, but after a while, in getting further insects visiting the plant doesn't actually increase the fitness of that, the fitness contribution of that flower to the plant's fitness. However, that might be quite different from the male's function point of view, because a flower can be visited more than once, and every time, and can be delivering pollen onto a series of pollinators, and each time a pollinator visits a flower and then leaves it to go elsewhere, there is a possibility for that plant to be transmitting genes to the next generation through siring seeds on other individuals. Now, in the end, the male and the female functions are equivalent, but this leads to very stiff competition amongst plants from their male function point of view in mating. This is effectively the plant's equivalent of sexual selection, and it leads to, I think, and in fact a great range of wonderful strategies that plants have evolved to maximize their fitness through their male function. So if we go back to this figure that I showed you a moment ago, most of those in those strategies that we can see here can be understood, I think, from the point of view of the difference between selection on male versus female function. And I won't go through all of them. We could give a whole lecture about that slide on its own. I'll pick out just two examples here. This one here is a lovely example which we've shown to students in the Iberian Peninsula. It's a Scrophularia species. It has 4 anthers, so each of these flowers is a bit like that, with 4 anthers that eventually come out. And the flower starts out with its stigma basically extruded from the floral tube, and then one at a time the anthers come out, one at a time over a period of days. And so you can see this flower here actually already has its 4 anthers, you can see them there, but actually younger flowers will only have 1 anther, then the 2nd anther, the 3rd and the 4th. What is happening there? That is a pollen dispensing mechanism, so that the pollen is not being dumped onto the first pollinator that comes along. It's increasing the possibility of siring to a number of different plants in the population. That is a very common strategy that we find in plants. This flower over here is another example, an Alpinia species in the ginger family. You can see the anthers there, and that little thing that's curved up underneath the top lip of the corolla, the upper petal there, is the pistil with the stigma at the top. How does this flower work? Well, that flower, this photograph, was taken in the morning. Pollinators enter the tube. Pollen is deposited on the back of the pollinators, bumblebees for example, and they then leave. But the stigma is up there, so those pollinators visiting flowers in that state will never be actually touching the stigmatic surface of the flower. At lunchtime, all the pollen has been removed, and the stigma now moves down. So the stigma in the afternoon is in the bottom position. Any pollinators visiting those flowers in the afternoon are going to be touching the stigma. There's no more pollen left in the anthers. And so you can see that that's going to lead to the potential for gene flow between flowers in this state and which is the male stage of the flower, and the female stage. Well, you might ask, how does that actually work? Because of course you've got to have flowers in both stages at the same time. But what happens is that there is a genetic polymorphism in these populations, and in the morning the other half of the population has their anthers held slightly further up, and the stigma is down. So the anthers don't open until lunchtime in the other half of the population. And so there is basically a single locus, probably a supergene, which has not been characterized, which determines the behavior of the flowers in terms of whether they happen to be morning female and afternoon male, or the other way around. And this is a strategy that is almost certainly evolved to avoid male-female interference, and indeed experimental tests have shown that if you actually manipulate these flowers carefully, you can demonstrate that that strategy increases siring success in particular. Another thing that's interesting to do with students is to let them loose into the field and ask them to look at particular plants without telling them too much what to expect. And so here's a couple of students who have come across a plant that happens to have a polymorphism. Some of the plants are hermaphrodites and some of them are male. It's quite a rare polymorphism, and we ask them then to think about how that polymorphism might be maintained. It's not entirely obvious when you just think about it. The hermaphrodites can do without the males. We don't need males in a hermaphroditic population, and yet there are males being maintained in those populations. And so the exercise we do in this particular case is take the students back to the classroom and then pose the question, what would be the predicted number or frequency of males in a population in which there are hermaphrodites? It's a simple evolutionary ecology question. Can you come up with a model that allows us to predict the frequency of the males in the population. And they're quite excited to do this because they've discovered this polymorphism themselves. They're interested in the fact that you don't actually need males in a hermaphroditic population, and yet they're there. So there's a sort of an excitement in the question. The modeling is challenging for them. It's a very simple model, but with a bit of help they come to an answer. They come to a simple prediction. So this is how the model works. We assume that males produce a certain amount of pollen more than the hermaphrodites do. So males are actually better males than the hermaphrodites are. They're producing more pollen. And we say that they're producing M times more pollen than the hermaphrodites say. We assume that mating is random in the population. And we assume that at equilibrium, the hermaphrodites and the males have equal fitness. So that's quite an important concept that the students learn, is that at an equilibrium, obviously the fitnesses of different phenotypes have to be equal. And that's useful in deriving the model. And I won't go through the derivation. As I say, it's quite easy to do, but the students, with a bit of help, and the students with a bit of help actually do end up with this, with the prediction of the sex ratio, and that's what the model spits out. And they can come up with this equation. So the sex ratio is given in terms of only the factor M. How many times more pollen do males produce relative to the hermaphrodites? Well, that's wonderful, because these are two things that you can easily estimate by visiting the field. So we go back to the field the next day. They can count the number of males in the populations, and there are lots of populations, so we put them in different pairs, pairs of students in different populations, and they all have to get the sex ratio, and then they take a sample of the plants and they count the number of male flowers, make a few assumptions about the equivalence of flowers on males and hermaphrodites and so on, and they come up with an estimate of M. So they work quite hard. It's quite tedious getting these data, but they're quite excited about it because they've got their own prediction that they've derived. And then we put the data together. That's the figure, that's the curve that they've derived, the prediction, the frequency of males as a function of the relative pollen production. That's our M term. And that's what they're hoping will be found for the populations. Each of them has found a different sex ratio and a different value of M, and the sort of expectation is it's going to, at least the prediction is that the points should lie on that curve. And here's the data from one of the years we did this project. Totally hopeless. And so it's very interesting to see how the students respond to this. And when I first did it with the students, I was so interested that I wrote a little article about it, because it sort of showed me how students think about data and theory. Because they were immediately, this didn't work out, we must have screwed up. And we get them to discuss what might have explained the difference between, why are there points here? And the response is always, we've somehow messed up. We possibly missed some of the males, we didn't count them properly, or the estimate of male flower production was different. Something was wrong with their methods. And they never suspect that something is wrong with the curve. And as a process, we go through a process of discussing their data, and gradually it dawns upon them that actually that curve, even though there's a simple expression, is a very abstract thing, and there are lots of deep assumptions in making that curve. And this is the real world. Sure, there's going to be some experimental error in estimating those points, but they might well be quite accurate, and that is what's wrong. And I think that's one of the things that we as scientists are often guilty of. We believe the theory too much, and we don't believe our data enough. And so we go through thinking about what might be going on in populations to give these other points, and we get hypotheses that one of the assumptions was that the population is randomly mating. What happens if the hermaphrodites are self-fertilizing to some extent? Well, you can sort of see that if the hermaphrodites are selfing, there will be a family of curves that are basically sort of almost parallel to that one going across to the right. It's going to be harder and harder for males to invade the population if the hermaphrodites are selfing. And so quite quickly, the students come up with their own sort of extension, a more complicated model that can perhaps be more general and explain the data that they've seen. And in fact, I've given this example because it sort of started us off on a whole research program in trying to understand the frequency of males in hermaphroditic populations. And the model that we've come up for the particular case that the students looked at is the following one. It's a meta-population model. And what we think is going on is the following, that you start off with an established population of males and hermaphrodites. And instead of just having single populations, we have a meta-population where the males and the hermaphrodites are colonizing other populations through dispersal. And so there is going to be a type of selective sieve here favoring the hermaphrodites as colonists because they can self-fertilize. The males might be really good at competing for siring success in an established population, but they're not going to be good at colonizing new populations. So most populations will be established by self-fertilizing hermaphrodites. Those populations then increase in size as a result of initially self-fertilization, but once the populations become large and dense, now they're competing with one another for outcrossing. In other words, here we expect the hermaphrodites to favor their female function because there's a lot of there's a lot of self-fertilizing inbreeding, so by a female by a sex allocation, and here the populations actually should favor increased allocation to their male function. Two things can happen. The hermaphrodites could evolve increased male function because they're now competing with one another, or that's the scenario that would be very nice for a male to invade because there's such an opportunity, all these hermaphrodites that are mainly producing seeds, an opportunity for males to invade. And so males invade the populations, and then quickly the gene for maleness spreads, and you end up with a population in which you've got males and hermaphrodites as in the initially established population. And in fact, that's exactly what you see in the field. This is a plot of populations of this species, Mercurialis annua. And you can see that basically you've got populations without males, perhaps recently established by colonization, and others in which the males are reaching frequencies up to about 45%, which is about what you'd expect in an outcrossing population at equilibrium, and everything in between. And often these populations are really quite close together. And so this is like a meta-population perspective on sex ratio evolution and the consequences of fluctuations between selection through selfing on the sex allocation and selection through outcrossing once the population establishes. And so there's a very dynamic system here where population sizes are fluctuating, the mating system is fluctuating, and selection will be shifting backwards and forwards, favoring increased or decreased male allocation in these populations. Okay, let's go back to this slide and ask another question. All of these plants are hermaphroditic. Most angiosperms are hermaphrodites, but not all of them. There are plenty of dioecious species with separate sexes. But why are most plants hermaphroditic? What are the advantages of being a hermaphrodite? And that's a question you can ask your friends. Why are plants hermaphroditic? And I ask the first-year students in our classes that question, and they always come up with the same answer, and perhaps the answer that initially occurs to you. And that is, they can self-fertilize. It's a big advantage. Plants are sessile. They might not have mates. They might not be able to get their pollen out. So there's a big advantage in being hermaphroditic. It's a good hypothesis, certainly one that we'll look at in a moment. But actually, most plants do everything they do to avoid selfing. And they have evolved all sorts of wonderful mechanisms to do so, which I'll illustrate in a second. Why do they try to avoid self-fertilization? Well, this is something that Charles Darwin investigated at great length. It's not known by many people who work on animals that Charles Darwin actually wrote 6 books about plants. He worked a lot on plants, and after he published The Origin of Species, he spent a lot of time in his glass houses at Down House doing experiments with plants. And one of the experiments he did, again and again and again, was comparing the fitness or the viability, the growth potential of plants that he hand self-pollinated versus outcrossed of lots of different species. And he wrote a whole book devoted to this. This is a couple of pages, a photograph of that book, and illustrating in this figure here the relative fitness. You might be able to see here that there are parallel lines here, a long line and a shorter line, and the shorter line here represents the decreased viability or vitality of plants that he had self-fertilized. So there's inbreeding depression in plant populations. We know this. It doesn't surprise us at all if we've done courses in population genetics. And that's probably why plants are avoiding self-fertilization, because if the reduction in fitness is sufficiently great, in fact, the critical threshold is 0.5. If the fitness of a plant that is self-fertilized is less than 0.5, it's definitely going to be better to maintain outcrossing. And so plants have, as I say, avoided many ways to avoid self-fertilization. There is temporal separation of the sexes, where the male and the female functions are actually active at different times. And I gave the example of Alpinia, where you've got both male and female first strategies in the same population. There is the spatial separation of the sexes. Well, of course, here we have spatial separation of the sexes within a flower. Here we have the spatial separation of the sexes into different flowers of the same individual. And this can be extreme, where we have the evolution of fully separated sexes, where all the male flowers are on some individuals and all the female flowers are on others, and they are obligately outcrossing in populations. And then finally we have what's called self-incompatibility, which means if you take pollen from a plant and place it on the stigma, so there is self-pollination, you don't see self-fertilization, because the pollen grains are unable to fertilize the ovules as a result of a molecular genetic incompatibility system. And so those are several ways in which plants have evolved to avoid selfing. And I wanted to focus in on two of these that are obligate outcrossing mechanisms. The first one, self-incompatibility, and the second one is diesi, where we have fully separated sexes between different individuals in these willows. So this is a self-incompatible brassica, and a dioecious willow. And the question I'd like to consider now is how evolution might take populations from one strategy to another, not between these two strategies, but from an obligately outcrossing strategy to a facultatively self-fertilizing strategy, and back again. So if we start with an obligately outcrossing strategy, and we then move towards a facultatively selfing strategy, what might be the and this has occurred many, many times in angiosperm evolution, what might be the reasons for that evolutionary transition? Well, there are three potential hypotheses, two of which have been emphasized a lot in the literature. One is mate limitation, and that's the basic undergraduate intuition that hermaphrodites that can self-fertilize would be an advantage if there are not mates available. So that would be an advantage. Similarly, if there are no pollinators available, if you can self-pollinate yourself and then be able to self-fertilize, that would be an advantage. And then a final idea, which we've been thinking quite a bit about, is that if the inbreeding depression becomes sufficiently low, then the penalty for selfing is somehow removed, and actually selfing could be quite a good strategy. So what would bring about the reduction in inbreeding depression in a population? Imagine a population that's got a lot of inbreeding depression because there is this sort of constant rain of deleterious mutations onto the population, or onto the genomes of the in a population, building up the genetic load. That's the origin of inbreeding depression, all these deleterious recessive mutations at low frequency, but at many, many loci. We need to get to a situation where that threshold is reduced below 0.5. And we've been considering ways in which that might take place. And just to sort of set this up, in a sort of a summary form. On the one hand, we have inbreeding depression that builds up in outcrossing populations. Because populations are outcrossing, homozygosity levels will be held low, and therefore deleterious recessive mutations are not expressed, and they can be maintained at low frequency in the population. As soon as the population becomes inbreeding, for whatever reason, those deleterious mutations become homozygous. They are then expressed, and they can be purged by natural selection. And so that can lead to the purging of the causes of inbreeding depression, the mutations giving rise to them. But the question is, how do you go from there to here? You can see there's sort of like, there are two attractors in a population. Either the population is going to be maintained as outcrossing and building up more and more inbreeding depression because it's outcrossing, or the population is already selfing, in which case it's just going to become more and more selfing. So we expect that population should evolve either to become more and more selfing or more outcrossing. How do you go from one extreme to the other? And one possibility is through a demographic process. And here comes this idea of colonization again. Plant populations are often highly dynamic, and I introduced the idea of a metapopulation which changes the mating system from one generation to the next. Another way that the levels of inbreeding can be changed is through range expansions. And so we looked into this from a sort of conceptual point of view. If we now plot the relative inbreeding depression in a population and consider a range expansion that starts on the left here and moves to the right, and then we measure inbreeding depression along this range expansion corridor. Imagine plants expanding from the south of Europe into northern populations through repeated colonization events. Two things happen. One is we get an increase in the frequency of deleterious mutations that fix just by drift. That reduces the fitness of the population. So average population fitness is expected to decline as a result of the range expansion. But critically, at the same time, these populations become more and more homozygous and lose their diversity, and in the end it makes no difference whether the plant is selfing or outcrossing. The progeny are basically identical. And so inbreeding depression is depleted. Hence, the lower inbreeding depression at the edge of the species range. And we tested that in Mercurialis annua again, the species that we've been using as a model. We sampled populations across the Iberian Peninsula from Morocco through southern Spain into the north, west and northeast, and we measured genetic variation, and we find that the southern populations have high genetic variation, the northern populations have low genetic variation, probably the result of this range expansion from the south to the north. And we asked, okay, what's the inbreeding depression in these populations here in the north compared to those in the south? And recent work that we've done on these range expansions in Mercurialis annua have shown that the range expansion has changed the allele frequency spectrum in these populations. So if we look at the frequency of fixed derived sites for non-synonymous mutations, these are mutations that might be under purifying selection, we find that they are actually at a higher frequency in the edge populations than in the core population. So there does seem to be an increase in the frequency of non-synonymous mutations at the range edge populations, possibly as a result of a range expansion as predicted by the simulations. This is a similar pattern now for mutations that are putatively of large effect, for example, those that bring about premature stop codons in exonic regions. And again, we find an increased frequency of those in the range edge populations than in the core, suggesting that there is an increase in the genetic load. And if we measure now inbreeding depression, we find now from the populations in Morocco and southern Spain quite high inbreeding depression for male reproductive effort and total fitness. And in the northern populations, inbreeding depression is low. And the question then is, can this whole process bring about the loss of an obligate outbreeding mechanism like self-incompatibility, for example? So I teamed up with Francisco Encinas, who's been working with me as a postdoc at the CSIRO in Australia, and we did similar simulations, but now we actually sort of advanced these a bit more to consider what happens to a self-incompatibility locus. And I won't go through all the details of the genetics of the locus. The important thing to state, just to start off with, is that we imagined a metapopulation in the species core that came to an equilibrium between mutations, selection, and drift. And we measured inbreeding depression, which tends to be quite high in these simulations. This is inbreeding depression here. And then we allowed the range expansion to occur. And as the earlier simulations had shown, we find that inbreeding depression does drop to quite low values. And those are the situations where we might expect self-compatibility to be able to spread in the population. So mutations are now occurring in the same species during this range expansion that are knocking out a self-incompatibility system, allowing plants to self-fertilize if they can. And so this is now an example of a simulation that shows not inbreeding depression, but the frequency of self-compatibility. And if we allow that simulation to progress, what we see initially is an expansion. Presumably these populations have now got low inbreeding depression, and it's in the range edge populations that self-compatibility invades. And we can allow that process to continue, and eventually we end up with a situation that leads to a type of stable species-wide polymorphism in which the range edge populations have become self-compatible and highly selfing, whereas the range core populations are still maintaining an outcrossing mechanism. And that's indeed exactly what we see in many species of plants that have variation in the mating system, that it's the sort of the core populations tend to be more outcrossing than the edge populations. Up until now, people have thought that that's because range edge populations tend to have low mate availability, maybe low pollinator availability. But actually, we think that another reason could be simply because patterns of inbreeding depression have been changed, allowing self-compatibility mutations to invade. Let's now think about breaking down the other obligate outcrossing mechanism, separate sexes, so plants that have evolved separate males and females. Sorry, I'm jumping ahead of myself. I wanted to just quickly illustrate an example from the real world of the breakdown of a self-incompatibility system, and then we'll go on to the dioecious case in a minute. Here is an example of what happens when self-fertilization does evolve, for example, in range edge populations. We see the evolution again and again in different species or genera of the evolution of self-compatibility and the so-called selfing syndrome, where plants produce smaller flowers, allocate less to pollinator attraction, etc., etc. And we decided to study this transition in a species that shows variation in the mating system between populations. This is the plant. It's a Linaria species found in southeastern Spain. Here are some of the populations we've been studying, and we find in the north here populations that are self-compatible, whereas the southern populations, and generally species, is self-incompatible. And we wanted to know, well, what's going on in these self-incompatible populations? And indeed, the question arose, once self-compatibility has fixed in a population like the COV population, can it then reinvade these populations, or invade these populations, and effectively spread throughout the species range as a result of migration and then selection? And so Marie, my PhD student who did this work, decided to address this by actually setting up experimental populations with mixtures of self-compatible and self-incompatible plants. The species in the wild has very high genetic differentiation amongst its populations. Here's the self-compatible population, it's an outcrossing population. And you can see that this is, a scatter of many other species, and Linaria happens to have very high genetic differentiation compared with most species. So it's highly, the populations are highly isolated. And so the question arises, given this high isolation, the mating system can evolve relatively independently within the populations, but you could then get migration from one population to the other. Could that bring about a species-wide spread of self-compatibility? And so that's the question. Could self-compatibility spread into the other populations and expand throughout the species range? And so this was the experiment. We set up 6 populations in which we have mixtures of self-compatible and self-incompatible individuals. And there are 3 pollinator treatments with 2 replicates in each. Total pollinator exclusion, so we put nets over the plants, so pollinators are excluded. Populations in which pollinators were allowed to access the plants at any time of the day or night. And then partial exclusion, where for a couple of hours, 3 times a week, the sides of these cages were opened up and pollinators could access the populations. And then we basically just sort of had a random passage of progeny from one generation to the next over 3 generations, and we wanted to track the mating system, various different other aspects of the biology of these experimental populations, and particularly the selfing rate and the frequency of the 2 different morphs. So the first thing is that the selfing rates for the self-compatible plants were 100% in the absence of pollinators. So if you exclude pollinators, the plants just self-fertilize. That's not surprising. Maybe slightly more surprising is that if you allow pollinators in, even though they can self-fertilize, they're actually outcrossing quite a bit. So if the pollinators do come visit the plants, they will outcross, even though they're self-compatible. It's what we call a species with a mixed mating system, where there's some selfing and some outcrossing. Then we looked at the numbers of fruits produced per flower in the self-compatible and the self-incompatible individuals, and we find that if there's no pollination, the self-incompatible plants generally produce no fruits. That's not a surprise. They can't self-fertilize. They're self-incompatible, whereas the self-compatible ones obviously do, and these are all self-fertilized, as we've just seen. When there is some pollination, then the self-incompatible plants start producing fruits, as you'd expect, but the self-compatible ones produce more fruits because they can both outcross and self-fertilize. And as a result of this over a period of time, we then looked at also the relative siring success of these, and this was the big surprise to us, that actually not only female fitness was higher for the self-compatible plants, but male fitness one was as well. So under low pollination, the self-compatible plants were siring most of the progeny, even on the outcrossing self-incompatible plants. This was somewhat reduced in the high pollination, but they were still siring most of the progeny. So they really are siring much more progeny than the outcrossed ones. And the net result of both male selection on male function and female function is that over a period of only 4 generations, we see almost the complete fixation of self-compatibility in these experimental populations, suggesting that perhaps in the wild the same process could happen with the invasion of self-compatibility into the populations, into the outcrossing populations. And when we did experiments to try and address this question of the relative siring success by putting pollen from outcrossing self-incompatible individuals and self-compatible individuals onto the same stigmas, and looked at the relative competitive ability of the pollen grains, we find that again, pollen produced by self-compatible plants was more effective at siring offspring than pollen produced by self-incompatible plants. That's not what we predicted, and we certainly don't understand this result. So we're still investigating this further. Why is it that those individuals that have evolved a strategy of self-pollination, or at least an ability to self-pollinate and self-fertilize, are actually better at siring outcrossed progeny as well? It's certainly not what the theory would predict, and not what we were expecting. Okay, finally I wanted to visit this question of the evolution of combined versus separate sexes. as I say, most plants are hermaphrodites, but separate sexed flowers and species have evolved many, many times. It's one of the most frequent transitions that has occurred in sexual system evolution in plants. And although dioecy separate sexes occurs in only about 6% of species, it's found in about half of plant families, suggesting that it evolves very often, but doesn't actually lead to massive diversification, and perhaps is an evolutionary dead end. Well, as I'll show, it's not an evolutionary dead end necessarily, and recent comparative work in Lyon has shown that actually it's much less likely to be an evolutionary dead end than people had hitherto thought. Here are a few examples of dioecious species, and if you compare those with that slide, something will strike you, I think, and that is that dioecious species are a lot duller and less interesting from their floral morphology and floral strategies than hermaphroditic species are. They tend to be wind pollinated, they have small white or green flowers, if they're insect pollinated, they're visited by generalist pollinators. They're almost never pollinated by birds, I think never pollinated by bats. They don't have specialist pollinating mechanisms. And the question is, what's going to bring about the transition from that sort of strategy, hermaphroditism, that seems extremely effective and successful, to a strategy that evidently has evolved but doesn't do so very often, and certainly doesn't lead to a great deal of diversification. And there are two hypotheses for the evolution of separate sexes. One is the inbreeding avoidance hypothesis that we've already addressed. It's another way of avoiding inbreeding. And the other is the sexual specialization hypothesis. And that one has received much less attention in the literature, but it's the idea that once separate sexes have evolved, the males and the females are now free to evolve a different ecology that would perhaps give them access to the resources that are available or required for their separate functions. Male function and female function might be really make quite different demands on the plant. And while plants are hermaphrodite, they're sort of stuck between these two. So if you're going to be a very good male, it might be best just not to abandon female function altogether, and vice versa. And so this is an example of a South African species in the genus Leucodendron, in which the males and the females look very different because they have different ecologies. So we're going back to this species Mercurialis annua now, and here is an example of a species in which we find both hermaphroditism and dioecy, another great system to address this sort of transition. Here are plants from a dioecious population. The females here, the males have this very interesting inflorescence which differs from the females, and the male flowers are held on these little stalks. It's a wind-pollinated plant, and what's interesting about this particular species is that in Spain and Morocco, as we've already seen, you find hermaphrodites where you've got female flowers surrounded by male flowers. The hermaphrodites are female-like in the sense that they don't have this excellent way of dispersing pollen. So there's a genetic secondary sexual dimorphism in inflorescence design between males and either females or hermaphrodites. We know that the males have a Y chromosome, so this is an XY sex determination system, and that the females and the hermaphrodites are effectively XX. Monoecy, however, is found in the polyploids. At high density, having your pollen dispersed from these long inflorescences is a real bonus in terms of siring success. A pollen grain dispersed from there has a 60% greater outcross siring success than a pollen grain dispersed from within the inflorescences down here, as in the hermaphrodites. So you sort of can see that if you're going to produce pollen in the species, you may as well be a male. And we've seen that perhaps the reason why these hermaphrodites are producing pollen is not really because they're very competitive for outcrossing, but because they can self-fertilize during colonization. Interestingly, pollen production is regulated through the expression of autosomal and X-linked genes, because these guys do not, these females and hermaphrodites do not have a Y chromosome, but they're producing perfectly decent pollen. What is on the Y chromosome is not what's needed for pollen production, it's the ability to produce that stalk. That is what's on the Y chromosome. And that, I think, is what we would call a sexually antagonistic phenotype that's great for a male, and it's probably not that useful for a female. We don't have estimates of the implications for females yet. And the male-like inflorescence is Y-linked in all of the species of Mercurialis that we've been looking at. At low density, so this is in a colonizing situation, the selfing rate is indeed density dependent, as the metapopulation model suggested. So at low densities, we've got, so these are the low densities, high interplant distances, selfing rates are very high. At intermediate and low interplant distances, high densities, you see there's much more outcrossing going on. Okay, just a second. So we've seen that there is a major functional implication for having separate sexes. From the male's point of view, it's a very good idea to do it well, to produce a morphology that is good at dispersing pollen. And yet we see populations in which hermaphroditism has evolved. And the question we posed was this the result simply of polyploidization? I mean, I've invoked selection in this metapopulation context, but can we actually show that this transition can be brought about by natural selection? Because we don't find hermaphrodites throughout a large part of the species range in Central and Western Europe. And so Guillaume Cossard, another PhD student of mine, basically tackled this question again by doing some experimental evolution with plants. And we basically started off with this simple observation, and that is that even though you might find populations of dioecious Mercurialis annua with females and males, and there's a strong sexual dimorphism, good sex chromosomes and so on, the females occasionally produce male flowers, and the males occasionally produce female flowers. And this is very common in plants. They are a type of they have a type of leakiness in their sex expression. They occasionally produce flowers of the opposite sex, even though they're actually quite good males and females, with all the other hallmarks of having separate sexes. And so we reasoned that this is the sort of variation that natural selection could grab hold of and bring about a transition from separate sexes back to hermaphroditism. Can leaky dioeci kickstart the transition back to dioeci? And how long would that actually take? And so this is the experiment that Guillaume did. He's again had 6 populations. In 3 populations, we had basically 50% males, 50% females, just as you find in the wild. And in 3 populations, we removed the males. And so now they're only females. And the only way these populations can reproduce at all is if some of the females produce pollen. And they do occasionally do that. And so the prediction would be that if these populations indeed manage to get over this bottleneck, where there might be very severe pollen limitation, which indeed there was, we predicted that natural selection would quickly increase the amount of pollen being produced by these individuals. And so we basically left these populations for a number of generations. We're now into the 6th generation. I'm going to show you data from the first 4 generations, which we've analyzed completely now. And we did a big common garden experiment. After 4 generations, we basically took the progeny from each of the generations and then grew them all up in a big common garden under the same conditions and measured the phenotypes from each of the generations and each of the different replicates, which is nice. You can do with that with plants because you can store the past generations. And these are the results, and it's quite remarkable, I think. The 4 generations, those are the data here in terms of the actual amounts of pollen being produced, this is the fold difference in pollen produced by females in the experimental lines without males compared to the lines that were just basically dioecious with males and females. And after one generation, females doubled their pollen production. After 2 generations, it went up to about 20-fold what they were producing after the first generation. And finally, after 4 generations, they're producing 50 times more pollen than they were at the start. So they have now evolved into this phenotype. That's what a normal female looks like. And this is one of the evolved females with all these male flowers. You can't really distinguish that from a normal monoecious individual. So in 4 generations, we've seen a transition to what looks quite similar to moneci that we see in Spain and the rest of the, Spain, Morocco, and Portugal. And so we now ask the question, to what extent has this very strong selection actually brought about a masculinization of the transcriptome, not just the phenotype as we measured in terms of pollen production, but the transcriptome? And there are not that many genes in the Mercurialis genome that have sex-biased gene expression. There are less than 1,000 genes out of the 28,000 or so that we've been able to assay that have sex-biased gene expression. About 900 of them, so a very low percentage of sex-biased genes, which contrasts with what we find in many animals where you've got very high levels of sex-biased gene expression, but it's quite similar to what we find in other plants that are dioecious, where sex-biased gene expression also tends to be quite low. So what Guillaume did was take 4 different phenotypes and looked at the amount of, and looked at the gene expression patterns. So we basically pooled 10 individuals, 10 females, 3 times, so we've got 3 different replicates, each of them pools of 10 females, 10 males from the control lines, the same again for females in the selected lines, because in the selected lines, although we've got these evolved phenotypes, we still see females segregating in these populations. So we've got a nice sort of comparison between the 2 lines through the females here, and then these evolved selected lines. And what we predicted was that if this is sort of like a cartoon of levels of gene expression where males and females differ in their patterns of gene expression, we might expect that after selection, the females shift their pattern of gene expression to something that looks a little bit more like the males do. So they're sort of moving across to the right because they're becoming more masculinized by natural selection. And indeed, that's exactly what we see. So this is the difference, this is the distribution of the difference in gene expression levels between the evolved females that are now producing a lot of pollen and the control females from the control lines. And this is for the, as I say, yeah, these are for the actual plants that are producing pollen as females, and there is a significant difference between these 2 distributions. The plants have indeed become more male, but more masculinized. The male bias genes are now overexpressed in the masculinized females, and we don't find that difference in expression between the unmodified selected females. So even though the unmodified selected females have experienced selection in recent past, their gene expression levels actually reflect the phenotypes that we measured. Okay, so that's all I am going to present now. What we see, just to sort of summarize, is that plant sexuality is very strongly shaped by selection on both the female and the male functions. And when you look at flowers in the field, think about that, because a lot of the adaptations that you'll see in flowers are probably there because they're enhancing a plant's ability to compete for siring success. If you take a time to just look at the different ways in which plants are exposing their pollen, you'll see lots of these strategies for yourselves. Another important point is to recognize that although most plants are hermaphroditic and self-incompatible, transitions to self-compatibility and selfing are occurring all the time, and often are being selected to the extent that they spread throughout the whole populations, bringing about a selfing syndrome, and that this can come about as the result of changes in the ecology because of mate limitation or pollinator limitation, but also changes in the genomic context because of demographic processes like metapopulation dynamics or range expansions. And indeed, in species that have a very fragile life history, the mating system is very dynamic, and selection is probably very strong in different directions in different populations. And so what we're actually seeing within populations often needs to be understood at a much broader sort of landscape level in those sorts of species. And then finally, in terms of how these transitions can actually take place, they can take place very quickly, as we've seen. There's a lot of standing genetic variation in populations that can allow populations to shift from separate sexes to combined sexes, back again, and so on. So if there's one sort of message, it's that plant mating systems are extremely dynamic, and things can happen quickly, and selection can be very strong and context dependent. So I'll just finish by acknowledging the people who contributed to the particular work I've presented here, and a few funders who've provided resources over the years, and thank you very much for your attention.